Good evening everyone. This video is going to be on the notes on the chapter 4 from the book by J V Forrester on Fundus Autofluorescence. So in chapter 4 we saw so many experiments on the rats and the mice to draw the conclusion about the lipofuscin formation and how we can stop it, because lipofuscin we have seen that it is cytotoxic, it is inflammatory, and it initiates and triggers the cell apoptosis as it accumulates in the RPE. It has got effects on the retinal pigment epithelial cells, which are seen as RPE degeneration and hence depigmentation and even release of the pigment, so hyperpigmentation in ARMD that we see because of the accumulation of the lipofuscin. So this fundus autofluorescence, it is a recent investigating modality in clinical ophthalmology. It's very important to understand the formation of the lipofuscin because it is the molecule that was found to be associated with the fluorescence and exhibiting the fluorescent property, and they have separated this A to E fluorophore from the lipofuscin, that is predominantly a prominently studied fluorophore. There are many fluorophores comprising the or contributing to the wide emission spectrum of the lipofuscin. So I'm just going to talk about these experiments in the animals in short. So first experiment was about CCR2 and CCL2. This CCR2 and CCL2 genes. Now every gene is responsible for production of certain proteins, and enzymes are nothing but the proteins. So various enzymes, various proteins, they regulate the cell function. So this CCR2 and CCL2 knockout mice means these genes were absent, CCL2 and CCR2, and this CCL2 is monocyte chemoattractant protein 1. So when these genes were absent in mice, so enzyme deficiency related to these genes, and what happened in these mice, they developed these ARMD kind of changes. They saw fundus autofluorescence in the eyes of these CCR2, CCL2 knockout mice. So they developed drusen, geographic atrophy, choroidal neovascularization. So these genes were found to be protecting against this ARMD, CCL2 and CCR2 and CCL2. These are protective genes. Then complement factor H gene, CFH gene. So this gene was absent in experimental mice, means then these mice are called as CFH knockout mice, like specific deletion of this gene in them. So complement factor H, this complement factor H gene is responsible for certain enzymes which regulate the alternative pathway of complement activation. Then, okay, so complement factor H gene, when it was absent in these mice, they found there were increased subretinal deposits and increased autofluorescence. Means this complement factor H gene, it has got protective function against ARMD. So which genes they found have protective functions against this ARMD? CCL2, CCR2 and complement factor H gene. Third experiment is about induction of the autoimmune uveitis in mice, how they caused. They injected this protein in them in mice, human interphotoreceptor retinol binding protein they injected in mice, and it caused uveitis, autoimmune uveitis in mice, and they saw the increased autofluorescence in these mice, and that was because of these macrophages. F480 plus macrophages. So the autofluorescence comes from the macrophages and RPE cells, and it is because of the lipofuscin. So these three experiments I talked about, out of which three genes were found to be protective against the ARMD, CHF, complement factor H gene, CCL2, CCR2, and they found that uveitis, increase in the autofluorescence in uveitis in mice, it was because of the macrophage, obviously the lipofuscin in macrophages. Then how the lipofuscin is formed. So we know that lipofuscin formation, it is because of this lysosomal enzyme deficiency, then or lysosomal dysfunction, then autophagy, and cellular stress. These three things are responsible for the lipofuscin formation. Another fourth experiment, they found that serum vitamin A levels have association with lipofuscin. How they found it? They saw the mice who were deficient in vitamin A. Uh, there was decrease in the fundus autofluorescence. So they found that vitamin A, that is related to the normal visual cycle, is responsible for the lipofuscin formation. So if the normal visual cycle is slowed down, the lipofuscin formation process is also slowed down, and hence the ARMD pathogenesis is also slowed down. The progression is also slowed down. So that was about serum levels of the vitamin A, and they also isolated the vitamin A dependent fluorophore in the lipofuscin using the thin layer chromatography. Then another thing, they injected leupeptin, which is lysosomal protease enzyme inhibitor. They injected this leupeptin enzyme, and it was responsible for increasing the autofluorescence and ARND kind of changes in the fundus. So lysosomal protease enzymes have protective function, means they are responsible for degradation or catabolism of this lipofuscin. So that was about the leupeptin, means lysosomal protease enzymes are important. They should be present, so there won't be any accumulation of the lipofuscin. Then another thing is RPE65 gene and its relationship to the lipofuscin formation. RPE65 gene, it is gene, so obviously it's going to produce certain proteins and enzymes of proteins. So these RPE gene proteins enzymes, they take part in the regulation of the visual cycle. They are responsible for the conversion of the all transretinal into 11-cis retinal. And if this RPE65 gene was absent, that is RPE65 knockout mice or knockout rat, they found that if this gene was absent, there was decrease in the formation of the lipofuscin. So this gene, if it is its presence, means normal visual cycle, and RPE65 gene absence means slowing down this or arresting this normal visual cycle and arresting the formation of the lipofuscin. So we can see which genes were protective. CCL2, CCR2, complement factor H genes. When they were absent, that led to ARND kind of fundus changes in the rats and mice. So first two experiments, then serum vitamin A levels, decrease in the serum vitamin A levels caused decrease in the lipofuscin formation. RPE65 gene antagonists, that we'll see later on, this RPE65 gene, when it is present, it is responsible for the normal visual cycle, and hence there is lipofuscin accumulation. And when this RPE65 gene was absent in rats and mice, in experimental rats and mice, that is RPE65 knockout mice or rats, their lipofuscin formation was decreased and ARND progression was slowed down. So all these studies indicate the normal visual cycle and serum vitamin A levels. These play important role in the formation of the lipofuscin. Now we know that how the lipofuscin is formed, like rough idea now, not like in a detailed way, but we have this much. We have done these many experiments, and we have concluded that there are vitamin A dependent fluorophores in the lipofuscin, and vitamin A and visual cycle is important for the formation of the lipofuscin. And lipofuscin is not just the consequence of the retinal degenerative diseases like ARMD and Stargardt's disease, but it is actually the cause of the disease. It is a causative cause. Actually, it is the cause of the disease. It is a causative factor for the disease progression and disease. So if we understand the formation of the lipofuscin, we can halt its formation, we can stop its formation or halt or slow down its formation, so we can slow down the progression of the disease, or we can erase the progression of the disease. That's the aim, to study all these things. That's the aim, to study the formation of the lipofuscin. So three drugs they have tested in animal models: isotretinoin, fenretinide, and RP antagonist, non-retinoid isotretinoid. So isotretinoin, how it acts, it slows the synthesis of 11-cis-retinaldehyde, and hence it slows the regeneration of the rhodopsin. When it slows this regenerative process, it slows down the visual cycle, and again it slows down in turn it slows down the lipofuscin formation. Now second thing, fenretinide retinamide, CHPR, NC4-hydroxyphenyl. So fenretinide, it causes a reversible reduction in the levels of the serum retinol, and when these serum retinol levels are decreased, as serum retinol is required for the visual cycle is slowed down, and again the lipofuscin formation will be slowed down. These all things were tested in the ABCA4 knockout mice. ABCA4, this gene is responsible for, actually this ABCA4 means ATP binding cassette protein A4, and it is responsible for the transport of these vitamin A in and out of the RPE and photoreceptors. So that is the function of this ABCA4 gene: transport of these metabolites and regulation of the visual cycle in RPE and photoreceptors. So if this gene was absent, and if these drugs were used, isotretinoin and fenretinide, they found there was reduction in the lipofuscin formation. Now they have also seen that it may promote, this fenretinide may promote the CNV complications in mice eyes, in ABCA4 knockout mice. May promote, they have mentioned it here. Then third drug, RP antagonist, non-retinoid isoprenoid. Non-retinoid isoprenoid, it is RP65 antagonist. So obviously RP65 gene and its proteins that it synthesizes, which act as enzymes to regulate the visual cycle, how they monitor or regulate visual cycle. They are responsible for conversion of the 11-trans-retinol to they are responsible for conversion of the all trans-retinol into 11-cis-retinol, and if this process is not occurring, or if it is slowed down, then it reduces the production of the lipofuscin. So these all drugs, they slow down the visual cycle. So visual cycle is slowed down means what? Light adaptation is slowed down, dark adaptation is slowed down. So we want the drug that has least effect on these adaptive processes, and more effect on the decrease in the lipofuscin formation. So these experiments are carried out in mice, and ABCA4 knockout mice, they have carried out these drug experiments. So that was the summary of chapter 4. So here I'll conclude this video. Next video will be on chapter 5 reading and chapter 5 notes. That I'll do tomorrow.